Proton pumps are protein complexes that move the protons generated during oxidation reactions across the cell membrane. As the protons move through the proton pump, they begin to build up on the outside of the membrane. The protons accumulate on the outside of the membrane, creating a concentration gradient. The membrane is not permeable to the charged hydrogen ions, and they cannot diffuse back across the membrane. Instead, they must pass through a special channel. The protons move through this special channel, which is the enzyme ATP synthase. This enzyme uses the energy derived from the movement of these protons to convert ADP into ATP. The movement of protons down a concentration gradient provides the energy for ATP synthase to form ATP. This mechanism of producing ATP is called chemiosmosis.
So let's finish up our discussion on Chapter 26. Let's have an overview of ATP production. Remember, we're talking about oxidative metabolism. So we have to consider the processes of glycolysis, the Krebs cycle, and the electron transport chain working as one large system to generate ATP to fulfill the energy needs of the cell. NADH releases an electron pair to the electron transport system, and protons are released then into the mitochondrial matrix to prime the pumps. So again, what we're looking at are the reactions that take place after glycolysis. This is once pyruvate has been passed into the Krebs cycle as a molecule of acetyl-CoA. So what's happening during the course of the Krebs cycle is that we're going to produce large amounts of NADH and FADH2 to act as carriers of high-energy electrons, which are then donated to the electron transport system. FADH then releases its electron pairs a little further along the electron transport system. The difference between FADH2 and NADH is that NADH produces enough energy by its oxidation in the electron transport chain to generate 3 ATP per each NADH oxidized, whereas FADH2 produces a grand total of 2 ATP for each molecule of FAD oxidized. So the electrons that are present here actually have more chemical potential energy, or more potential energy, than the electrons that are released from this particular carrier. The complete aerobic oxidation of glucose to CO2 and water plus ATP and heat produces between 36 and 38 ATP per glucose molecule. And this overall efficiency is about 40% of the total chemical potential energy contained in a molecule of glucose. The rest is lost as body heat. Remember that every transformation, every energy change loses a little bit of that energy as heat to the universe. This is the second law of thermodynamics at work. Okay, what we're looking at here is just an overview of the oxidation of glucose. We want to separate where these reactions take place. Notice that glycolysis takes place primarily in the cytosol. The net production of ATP in glycolysis is 2 ATP per molecule of glucose, and the net production of NADH from glycolysis is 2 NADH per molecule of glucose. The end product of this particular metabolic pathway is pyruvate. The pyruvate is then, if oxygen is present, passed into the citric acid cycle, where it will be utilized to form directly 1 ATP per pyruvate molecule, so 2 ATP per glucose, 1 FADH2 per pyruvate molecule, so 2 FADH2 per glucose molecule, and 3 NADH per pyruvate molecule, equaling 6 NADH per glucose molecule. Now what happens to the NADH and the FADH2? Well, they're oxidized in the electron transport chain. Two things happen. These electrons are passed to carriers that are embedded in the intermitochondrial membrane. And as those electrons lose energy, that energy is captured and used to pump protons from the matrix of the mitochondria into the intermembrane space. Finally, as those protons flow back down their chemical potential gradient from the intermembrane space to the mitochondria, that energy is captured by an enzyme called ATP synthase, and that's where the bulk of ATP production takes place. You get about 32 ATP as a result of the complete oxidation of the NADH and the FADH2 produced by glycolysis and the Krebs cycle put together. So the net ATP produced is shown here. You get 4 ATP directly off each of the cycles, and you get between 28 and 30 ATP as a result of ATP produced in the electron transport chain, specifically by ATP synthase. So that gives us a grand total of between 36 and 38 ATP per molecule of glucose oxidized. And you can see how much more energy that is per molecule than the anaerobic portion of this pathway, which is entirely up here, contained in glycolysis, where you get a whopping 2 ATP per glucose molecule. So you're talking almost 20 times more energy in the presence of oxygen than you get with simply running glycolysis. And that's why the cell can't meet its energy needs for an extended period simply running on anaerobic respiration. This is why we need oxygen, and we get the oxygen in the air that we breathe. Okay, let's talk about our energy stores. ATP is used after it's formed, and it's not stored. It wouldn't be practical to store it because it would take up too much physical space and it would break down after a while. Extra glucose won't be oxidized. Instead, it will be stored. It can be either converted to fat or it can be used to synthesize glycogen. And this process is called glycogenesis. It's stimulated by the hormone insulin. Glycogenolysis is the hydrolysis of glycogen to glucose, and it's stimulated by glucagon, which is actually an antagonist to insulin, as well as epinephrine, also known as adrenaline. Only hepatocytes can release glucose into the blood under the stimulation of these two hormones. So this is the body's response to an energy deficit. These hormones are secreted. Glucose is then put into the blood, and it's delivered to cells that need it for energy. So you could think of these hormones as the delivery man, the energy delivery man. Gluconeogenesis is the synthesis of glucose from non-carbohydrates such as fats and amino acids. And again, the liver carries this out. You can think of the liver basically as the body's kitchen. And what it does is it produces molecules that the body needs for energy as our energy demands change. And so let's take a look at how glucose moves around the body as energy needs shift. Here you can see a graph showing you how glucose is used and stored. So let's take a trip from glucose in the blood to energy in the cell. So blood glucose is delivered to metabolically active tissue, and the enzyme hexokinase produces glucose 6-phosphate by transferring a phosphate group from ATP to glucose. This traps the glucose inside the cell, and from there it can either enter glycolysis and from there eventually the Krebs cycle, or it can be isomerized into glucose 1-phosphate, and from there the enzyme glycogen synthase can be used to generate glycogen, which is a glucose polymer from monomers of glucose 1-phosphate. Let's follow glycogen on out to blood glucose. If we need glucose in the blood, one of the ways that we can do this is to hydrolyze glycogen with the help of an enzyme called glycogen phosphorylase. This produces monomers of glucose 1-phosphate, which can then be isomerized into glucose 6-phosphate, which can then be released into the blood as simple glucose through the action of glucose 6-phosphatase, which is found in the liver, the kidney, and the intestinal cells. And so this is what would happen if the body needed an immediate burst of energy from our stored reserves, as we could call on glycogen and very rapidly put glucose back into the blood. By the same token, if this glycogen were needed internally by the cell in which it was stored, the glucose 1-phosphate could enter glycolysis directly, and from there enter the Krebs cycle, and from there we would be able to produce the 38 to 36 ATP per glucose molecule that could meet the cell's energy needs. Now, we have to be conscious of the fact that not all these enzymes are present in all cells. The areas in the body where you're most likely to find glycogen stores are muscle tissue and the liver. So when you're talking about mobilizing glycogen, these enzymes, phosphorylase and synthase, are going to be expressed primarily in these tissues. The rest of the tissues of the body are going to rely on the mobilization of these reserves to deliver glucose into the blood and, as a result, energy to the cells that need it. Okay. Lipids come in many forms. Triglycerides are one, and they are stored in fat cells, which are called adipocytes. There's a turnover of molecules every 3 weeks. It's released into the blood, transported, and then either oxidized or redeposited in other fat cells. Lipogenesis is the synthesis of fat from other sources. Amino acids and sugars, for instance, can be used to make fatty acids and glycerol. So this is why you can eat something high in sugar and end up storing the excess calories as fat, because we're able to synthesize the components of triglycerides because of the enzymes that are present in, among other places, the liver. Lipolysis is the breakdown of fat for fuel. Remember that the body's ultimate energy store is our fat stores, our adipose tissue, and glycerol can be released from triglycerides, which are the monster's percentage of our fat that's stored in our body. This can be converted to phosphoglyceraldehyde, whereupon it can enter glycolysis. And remember that the fatty acids can be broken down in 2 carbon units, and these 2 carbon units can be used to generate acetyl-CoA, which can then enter the Krebs cycle and can serve to produce a lot of NADH and FADH2, which can then be oxidized by the electron transport chain to produce 32 ATP per acetyl-CoA that enters the chain, and the result then is a lot of ATP for a single fatty acid that might be, say, 18 carbons long. Remember that in 2 carbon units, that means you could produce 9 acetyl-CoAs and send each of those through the Krebs cycle and produce the necessary energy to supply the body's needs. So this is why fat is by far the most calorie-dense molecule in the food that we eat. And here you can just see a look at how lipids flow through the body. The green arrows indicate lipolysis, so this is the mobilization of stored triglycerides into energy for the body to use, and lipogenesis is the production of fat from non-carbohydrate or carbohydrate precursors, either one. So if we look at fat mobilization, for instance, let's say the body needed a lot of energy, we could call on our stored triglycerides, and the liver would be the place where we'd most likely start, and we could hydrolyze the triglycerides into glycerol and fatty acids. As we've stated, the glycerol can then enter glycolysis. From there, it can enter the Krebs cycle, and the fatty acids can be cleaved through a process called beta oxidation that produces 2 carbon acetyl groups. These can then enter the Krebs cycle as acetyl-CoA, and in the event that we incompletely burn our fat, we have more acetyl groups than we can actually send through the Krebs cycle. Part of this can form these molecules that are called ketone bodies. Examples are beta-hydroxybutyric acid, acetoacetic acid, and acetone. And these are blown off as we exhale. So this is why individuals that go on high-fat, low-carb diets tend to burn their fat incompletely because they lack sufficient Krebs cycle intermediates to deal with all this acetyl-CoA. And so essentially we're wasting energy. We're just exhaling it into the atmosphere. Now, if we look at lipogenesis, what we observe is that in glycolysis we produce phosphoglyceraldehyde as one of the intermediates. This can be oxidized to glycerol, and this glycerol serves as the backbone for the triglyceride. The fatty acids can be assembled from acetyl-CoA subunits, and then the fatty acids and the glycerol together can be ligated. Basically, they can form covalent bonds with each other, and this will generate a novel triglyceride. If you are interested in what the structure of a triglyceride looks like, refer to the back of your textbook, look up triglyceride, and there should be a figure of it in this chapter. And you can see that what you have is glycerol, which is an alcohol that contains 3 hydroxyl groups, and it's attached to 3 separate free fatty acids. And so what you produce is actually something called an ester in biochemistry. And this is a very rich store of energy because particularly the fatty acid side chains have a lot of chemical potential energy because they have a lot of carbon-hydrogen bonds. Okay, ketogenesis takes place when we're making more acetyl-CoA than the body can actually combust. Fatty acids are catabolized in the mitochondrial matrix by beta oxidation, and these acetyl groups can enter the citric acid cycle as acetyl-CoA. However, if you produce too many acetyl groups, the liver can turn around and perform ketogenesis. This generates ketone bodies. If the body is rapidly oxidizing fats, ketones can build up, and this can lead to ketoacidosis. And we talked about the fact in class that this is not a good long-term solution to fulfilling our energy needs because the body needs to maintain a pH near neutral in most tissues in order for enzymatic reactions to happen at any kind of appreciable rate. So ketoacidosis is not something that we want to have our body in long term. Okay, let's talk about protein for a little bit. Dietary amino acids and tissue protein are broken down every day into free amino acids. These can be used to make new proteins. They can also be used as fuel, but to do that they have to be deaminated. You have to remove the nitrogen group on them. What remains is a keto acid that can be converted into intermediates that you find in the Krebs cycle. In an amino acid shortage, the reverse takes place. We basically rob the citric acid cycle of intermediates to produce amino acids in order for us to be able to build the proteins the body needs to live. The ammonia that is stripped from these amino acids can become toxic if it builds up to high levels in the blood. What the liver does to combat this is it takes these amino groups and along with CO2 produces molecules of urea, which are excreted in the kidneys in the urine. This is our nitrogenous waste product. So we always want to be conscious of the fact that every time we take in protein, we're taking in amino acids, and we have to be conscious of the nitrogen balance. We always want to be taking in as much nitrogen as we're eliminating. Otherwise, we're going to have a problem. If we're taking in more nitrogen than we're eliminating, that could mean one of two things. It could mean that the kidneys aren't doing their job, or if we're a bodybuilder or growing rapidly, it could mean that we're synthesizing a lot of new protein. But if it's a result of the kidneys starting to shut down or for some reason not working the way they should, then the urea can build up in the interstitial fluid and in the blood, and that can kill you. And that condition is called azotemia, which can lead to uremia, which can lead to your death. And so here you're looking at some of the ways that amino acids, proteins, and keto acids, the Krebs cycle and glycolysis are all related. And so they're just showing you that we can move these molecules between these metabolic pathways because of enzymes that are capable of either deamination or transamination in order to generate, for instance, amino acids that the body needs to build protein, or say keto acids that the body needs in order to properly run glycolysis and the citric acid cycle. The back end of this is the urea cycle, and this is what generates the urea that the kidneys excrete, and this takes place again in the liver. So the liver does a lot of good work for you. It's your detoxifier, and here you're looking at the urea cycle. It uses ammonia to generate urea, which is removed from the blood by the kidneys, and you can see that there are some intermediates in the urea cycle that can be used to synthesize, among other things, proteins. Arginine, for instance, is an amino acid, and it's also an intermediate in the urea cycle. So the body has different ways that it can generate the non-essential amino acids necessary to fulfill the body's protein needs. Remember that the essential amino acids the body isn't capable of synthesizing, so we got to consume those in our diet. So it's in the food that we eat. And if we have all the essential amino acids in our diet, we're eating what's called complete protein. Animal protein is complete protein as is, whereas if you're on a vegetarian diet, you have to eat combinations of vegetables in order to have complete protein. Examples on a vegetarian diet would be, say, beans and rice would contain all of the necessary amino acids that the body needs to build the enzymes and the structural proteins and the antibodies that the body needs in order to function. Okay, so we've talked about the liver. We've talked about its role as the body's kitchen. Let's go into a little bit more detail. A variety of roles can be assigned to the liver. It acts in carbohydrate, lipid, and protein metabolism. Most of its functions are non-digestive. Hepatocytes perform all the functions except for phagocytosis. As a result, degenerative liver disease such as hepatitis or cirrhosis or even liver cancer can be life-threatening. In fact, the liver gets its name from the first four letters, live. It allows you to live. Without your liver, you wouldn't last very long because you couldn't detoxify compounds in the blood, you couldn't form nitrogenous waste, and you'd be in a very bad way in a short period of time. Some of the conditions that affect the liver are hepatitis and cirrhosis. Hepatitis is basically a clinical term for liver inflammation and is caused by 5 strains of viruses. Hepatitis A is common and relatively mild. It causes about half a year of illness in most people, but you generally recover. Hepatitis B and C are transmitted sexually through blood and body fluids. This can lead to cirrhosis of the liver or even to liver cancer. And you might ask, well, how is it that viral infections can lead to cancer? What you have to remember about viruses is that the way they propagate is that they introduce their genetic material inside normal cells. And that genetic material can migrate to the nucleus and integrate into the DNA. And if it integrates into a gene that controls the cell cycle, basically the instructions that tell a cell when and where to divide, the cell can basically go off script and start dividing on its own schedule, irrespective of the body's needs. So very often, certain types of cancers have been closely correlated with certain types of viral infections because we're altering the DNA. Cirrhosis is irreversible inflammatory liver disease. This is not something that you want to acquire. It results in the formation of scar tissue. The vessels that supply blood to the liver can rupture and necrosis can occur. It can result from alcohol abuse. It can result from recreational drug use. Remember that your liver is your body's detoxifier, and if you overload the liver, what you can do is basically cook the cells that are responsible for detoxifying your body's fluids. If And you might ask, well, gee, how does that happen? Well, think about it in terms of workload. You can overload a cell if you demand too much of it. And what can happen is that the cell can work to keep the body going to the detriment of its own survival. For instance, if a detoxifying cell is so busy detoxifying things, it may not have time or resources to generate ATP to keep the cell alive. Okay, so that's one way, for instance, that a cell could wear out. Okay, let's talk about the states that exist before and after a meal. The absorptive state lasts about 4 hours during and after a meal. This is where nutrient absorption fills our energy needs. Carbohydrates are basically the sugars that are available in the blood for all cells to use for ATP synthesis. The excess carbs are converted by the liver to glycogen or to fat, most of them to fat. Remember that there are only a few cells that store massive amounts of glycogen. What happens to the fats? They're taken up by the fat cells, the adipocytes, from chylomicrons that they find in the blood. And they're a primary energy substrate for liver fat and muscle because they are so calorie rich, because they have so many carbon-hydrogen bonds. Amino acids have to pass through the liver and then go on to other tissues. In hepatocytes, they're used for protein, ATP, or fatty acid synthesis. So remember that amino acids are the building blocks of proteins, so obviously they're important for anabolism, the building of macromolecules from smaller precursors. But we also have to be conscious of the fact that amino acids can serve as intermediates in glycolysis and the Krebs cycle. And so in that manner, they can facilitate metabolism. They can facilitate breakdown of macromolecules to provide the body with sufficient ATP in order to operate. Okay, how do we regulate the absorptive state? The absorptive state is regulated by insulin secreted in response to elevated blood glucose and amino acid levels, as well as in response to gastrin, secretin, and cholecystokinin, abbreviated CCK. These are digestive hormones that control whether or not we're going to store energy or whether we're going to mobilize energy and deliver it to metabolically active tissue. Insulin increases cellular glucose uptake and it stimulates glucose oxidation. Glycogenesis and lipogenesis are also under the influence of insulin stimulated, but it inhibits gluconeogenesis, the formation of sugar from non-carbohydrate precursors, and it also stimulates the active transport of amino acids into cells along with protein synthesis. High protein, low carbohydrate meals stimulate the release of insulin and glucagon, and this prevents hypoglycemia. Again, we want to keep sugar levels within homeostatic limits in the body. We don't want them to go too high for too long or too low for too long. And these hormones act opposite each other. Insulin lowers blood sugar while glucagon raises blood sugar. And so often these hormones are secreted together to keep glucose in the blood within a very limited range. Okay. The post-absorptive state is what happens long after a meal is over. The homeostasis of blood glucose is critical to brain function because the central nervous system can only use sugar as fuel. When the stomach and the small intestines are empty, we utilize our stored fuels. If we're talking about carbohydrates, we're talking about sugars, particularly glucose, which is drawn from the body's glycogen reserves for about 4 hours and then made from other compounds in the process of gluconeogenesis in order to deliver sugar to tissues that can only use that as a fuel source. Other tissues have other options. They can use fat, for instance, as a fuel store. Fat cells and liver cells can convert glycerol into glucose, and the free fatty acids can be oxidized to the liver and generate ketone bodies. The majority of tissues use fat for energy, and this leaves glucose as the major fuel for the nervous system. Now, protein, as we've seen, can act in a variety of ways. It's used when glycogen and fat reserves are gone. So in a starvation scenario. Wasting away occurs in the process of diseases such as cancer due to appetite loss and an altered metabolism. Essentially, what happens is that the fat stores can be used up by the rapidly dividing tissue in order to feed the growing tumor mass. And then once the fat reserves are gone, the tumor still wants to eat, it wants to live. And so what it'll do is begin to take advantage of the body's stored protein after it uses up all the fat. And this is why individuals who are in late stage cancer, say stage 4 for instance, often have a skeletal or skin over bones appearance because they've lost a lot of their fat reserves, and as a result their features take on sort of a gaunt appearance. So I wanted to talk to you about the regulation of the post-absorptive state. We are at slide 73 in the PowerPoint presentation on nutrition. First of all, I want to mention that it's regulated by sympathetic nervous system and glucagon secretion, which is generated by the islet cells of the pancreas. Remember that glucagon acts as a counterpoint to insulin. It mobilizes glucose and as a result makes that form of chemical potential energy readily available for metabolically active tissues by secreting free glucose into the blood. When blood glucose drops, glucagon is secreted. This results in glycogenolysis, which is the breakdown of glycogen into glucose monomers, along with gluconeogenesis, which increases free glucose levels. Remember that gluconeogenesis is the production of glucose monomers from non-carbohydrate precursors. So again, the effect is to increase plasma blood glucose. In addition to this, both of these types of innervation increase lipolysis, which is the breakdown of free fatty acids from triglycerides. So the net result is to increase the amount of chemical potential energy available as a fuel source for cells that need it for metabolic energy. So it's a good thing that we have this type of regulation. There are also sympathoadrenal effects. These promote glycogenolysis and lipolysis during injury, fear, anger, and stress. Examples would be adipose, liver, and muscle cells, which are richly innervated and respond to epinephrine. As a result, they're going to more easily break down these fuel stores into readily available consumable monomers in the blood plasma so that we can feed metabolically active tissue. Also, cortisol promotes an increase in blood glucose, and this is aided by fat and protein catabolism as well as gluconeogenesis, which again, remember, is the synthesis of glucose by the liver from non-carbohydrate precursors. In addition, growth hormone stops rapid decreases in blood glucose, which is tantamount to saying that it increases plasma blood glucose levels in order to, again, respond to increased metabolic need. Okay, let's talk about metabolic rate for a bit. Metabolic rate is the amount of energy used in the body per unit time. It's measured in a calorimeter or by oxygen consumption. A calorimeter is a device that contains the substance to be analyzed. It could be a chemical compound. It could be almost anything, really. This is then burned inside a ceramic chamber, and the amount of heat generated is measured in calories. So this is a direct measure of the amount of chemical potential energy contained in anything that you can manage to combust, to burn up. It can also be measured by oxygen consumption. Remember that oxygen is the terminal electron acceptor in the electron transport chain. It is converted to water as it accepts low energy electrons from the electron transport chain in addition to 2 hydrogen atoms to generate H2O. Basal metabolic rate is measured when an individual is relaxed, awake, fasting, and at a comfortable temperature. The adult male basal metabolic rate is 2,000 kilocalories per day. This is slightly less in females, owing primarily to differences in testosterone levels between the two sexes. Factors that affect total metabolic rate are pregnancy, anxiety, fear, eating, thyroxine, and depression. Again, these are all stimuli that will produce chemical messages inside the body that are the result either of nervous innervation or of endocrine secretion The target tissues will then respond with a change in metabolic rate, which will alter the rate at which they consume chemical potential energy to produce ATP, which is then hydrolyzed to do cellular work and fulfill cellular functions. Okay, thermoregulation and body heat. Homeostasis requires that heat loss is always equal to heat gain. Hypothermia is an excessively low body temperature. This slows metabolic activity and can cause death. Hyperthermia is an excessively high body temperature. This can disrupt metabolic activity and cause death. And let's think about the reasons why for a minute. Hypothermia is an excessively low body temperature. Chemical reactions drop by a level of 2 in their rate for every 10-degree drop in temperature. Eventually what would happen is that chemical activity would cease in terms of being able to supply needs that the body has for chemical reactions that occur rapidly enough to maintain life. Hyperthermia, on the other hand, would result eventually in the denaturation of enzymes that modulate metabolic processes. As a result, numerous processes would shut down and the organism would die. Thermoregulation is the ability to balance heat production and loss, and hopefully as long as we have thermoregulation, then we are fully capable of maintaining homeostatic levels in body temperature. Okay, body temperature. Normal body temperature varies about 1.8 degrees Fahrenheit in a 24-hour cycle. It's low in the morning and high in the late afternoon. Core body temperature is called the visceral temperature. This will be the temperature of the internal organs. Rectal temperature and axillary temperature can be used as estimates. Adults vary from 99 to 99.7 degrees Fahrenheit in these temperature variations. Shell temperature is the temperature closer to the surface, so the temperature on the skin. The adult varies from 97.9 degrees to 98.6 degrees Fahrenheit. If we're looking at heat production and loss to maintain homeostasis of body temperature, most production is from nutrient oxidation and ATP use. It mostly comes from the brain, the heart, the liver, the endocrine system, and the muscles. Exercise can increase muscular heat production to maintain homeostasis. The modes of heat loss vary. Radiation is body heat loss to the objects around us, and it's caused by molecular motion within the body, which produces infrared radiation, which is lost to the surroundings. Conduction is heat loss to the air by convection. Again, this is movement of molecules of gas with other molecules of gas, resulting in loss of kinetic energy. And evaporation is heat loss to water as sweat evaporates. So as water goes from one phase of matter liquid to another phase of matter gas, kinetic energy leaves the surface of the body. We sense that as a loss of heat, and as a result, homeostasis is maintained by cooling down body temperature. Under extreme conditions, you can lose up to 4 liters of sweat per hour. It's quite a bit. Okay, thermoregulation and negative feedback. The hypothalamus thermostat monitors the temperature of the blood in the skin. Remember that the hypothalamus is a structure at the inferior portion of the brain that lies just superior to the pituitary gland and controls the secretion of hormones and the production of hormones in this organ. So it's a very important controller of the endocrine system. And one of the things that the endocrine system controls is thermogenesis. So it signals heat-losing centers. If our body cools, the hypothalamus will pick this up when body temperature is high, and this will result in cutaneous vasodilation to promote heat loss. So we will begin reddening at the surface of the skin, and this is blood moving to the surface and cooling by convection and conduction. This triggers also sweating, which will also increase heat loss as a result of evaporative cooling. It signals the heat-promoting center in the hypothalamus when the body temperature is low. This causes cutaneous vasoconstriction, rerouting blood away from the surface to the core. And it stimulates the erector pili muscles to make the body hair stand on end, which creates dead air space between the surface of the skin and the external environment and reduces heat loss even further. so that our body temperature will begin to rise. It also triggers shivering and thermogenesis through skeletal muscle contraction that hydrolyzes ATP and produces as a side product heat, which will increase body temperature. And it also triggers non-shivering thermogenesis, such as an increase in thyroxine, which results in an increase in BMR. And if you remember from the lab we just did with the mouse that received the thyroid extract, which contains thyroxine, had the higher metabolic rate, and as a result was able to generate more body heat per unit time on average as a result of increased metabolic demand. There's also behavioral control of thermoregulation. We try to get out of the sun or remove heavy clothing if we are hot, and as a result we cool down. So all of these things play into maintaining homeostatic body temperature. There are also disturbances of thermoregulation which we should address. Fever is a normal protective mechanism that elevates basal metabolic rate, which produces more heat, leading to an increased basal metabolic rate. Hyperthermia is exposure to excessive heat. It can result in heat cramps, which are spasms due to electrolyte imbalance from excessive sweating. It can also result in heat exhaustion, which is severe electrolyte imbalance, and this can produce fainting, dizziness, and hypotension. And finally, it can result in heat stroke, which occurs when the body temperature rises excessively and can cause coma, convulsions, and death, none of which are a desired outcome. Okay, hypothermia is the other extreme. It's an exposure to excess cold. As the core body temperature begins to drop, the basal metabolic rate will drop, and this causes a further body temperature decrease. It can be fatal if the body temperature decreases below 75 degrees Fahrenheit, because then metabolic reactions are not proceeding at a rate sufficient to maintain life. So this is a not desired outcome as well. Okay, we want to talk a little bit about alcohol and alcoholism and its effects on nutrition, behavior, and physiology. Alcohol is a mind-altering drug. It's essentially empty calories. It can also be addictive and is a toxin. It has an LD50, as do other substances, but its LD50 is a bit lower than a lot of other substances we take into our body that we don't consider to be toxins. It's rapidly absorbed from the gastrointestinal tract. 20% of it is absorbed in the stomach and 80% in the small intestine. Carbonation can increase this rate. As a result, beer can be absorbed more readily than wine or spirits, liquor, and such. Food can reduce the absorption rate because the alcohol will essentially associate with the food molecules and as a result absorb through the stomach wall and the small intestine lining a lot more slowly. It can cross the blood-brain barrier to exert its effects on the brain because it is a small molecule and somewhat nonpolar, so cell membranes don't present quite the barrier to its diffusion across different portions of the physiology as they would present to a polar molecule or a molecule of significant size. It's detoxified by the hepatic enzyme alcohol dehydrogenase, which generates acetaldehyde, which is essentially a pickling agent that we'll address in a little bit here. This molecule can enter the citric acid cycle and can be oxidized to CO2 and water. Women have less alcohol dehydrogenase than men, and as a result are more vulnerable to alcohol-related illnesses and addictions. It's also found that certain ethnic groups have a hereditary lack of alcohol dehydrogenase, and as a result, alcohol addiction is more of a problem in these ethnic groups than in ethnic groups that have more of this particular enzyme in the liver. Okay, there are physiological effects to this addiction. It has an effect on the nervous system. It's a depressant which inhibits norepinephrine release and disrupts GABA receptors. Low doses can produce euphoria, and high doses cause the nerves to be less responsive to neurotransmitters. You've all been familiar with the effects of alcohol at high doses, either in someone you've observed or perhaps you've taken too much alcohol in yourself at some point during your life. Timing and coordination become impaired, slurred speech results. Also, poor coordination and slow reaction times can be an outcome. The liver is the primary detoxifier of alcohol, and excessive acetaldehyde buildup can cause inflammation of the liver and pancreas, and you can essentially pickle this organ. So when you've heard individuals say that they've pickled their liver due to massive alcohol consumption, they are being actually biochemically quite accurate because the action of this particular compound is similar to the action of formaldehyde on living tissue. So keep that in mind the next time you hear about someone imbibing too much in spirits to the point that they affect their health. It can destroy hepatocytes and produce cirrhosis, and this can result in hepatic coma and jaundice, none of which are desirable. Okay, further physiological effects. There are effects on the circulatory system. Clotting problems can occur because of factors becoming depleted due to liver damage. You can also experience edema due to inadequate production of albumin by the liver, and cirrhosis can obstruct hepatic portal circulation and as a result lead to inadequate cleaning of the blood and removal of waste by the liver. Also, portal hyper or hypotension can result. Hypertension will result in hypoproteinemia, where the liver and other organs will weep a serous fluid into the peritoneal cavity. Ascites is an extreme example of this. It's an abdominal swelling with several layers of this serous fluid. It can also lead to bleeding and hematemesis, which is vomiting of blood. And finally, it can destroy the myocardium, decrease heart contractility, and produce cardiac arrhythmia, all of which are undesirable outcomes. Further effects, it can affect the digestive system and your nutrition. It breaks down the protective mucous barrier of the stomach, which can result in gastritis and bleeding. Can also be linked to esophageal cancer and peptic ulcers. We discussed peptic ulcers earlier in this chapter, the fact that inflammation of the wall of the stomach can eventually lead to a structural breakdown in that particular organ, which would result in the leakage of gastric juice into the peritoneal cavity in the event of a perforated ulcer, which would result in the digestion of the contents of the peritoneal cavity, and this would be a very undesirable outcome. So something to be avoided at all cost. Alcohol can also result in malnutrition from suppression of the appetite, and in addition, acetaldehyde can interfere with absorption of certain vitamins and their use. So all of these are undesirable effects. We can also move into addiction as a result of overindulgence in this particular compound. It's the most widely available addictive drug in America, and it's similar in barbiturates to its toxic effects. There's a potential for tolerance, dependence, as well as a risk of overdose. You can acquire physiological tolerance at increased concentration, and this can result in impaired physiological, psychological, and social function. There are withdrawal symptoms when use is halted and intake is stopped. These are often referred to as DTs or delirium tremens. The symptoms are restlessness, insomnia, confusion, irritability, tremors, incoherent speech, hallucinations, convulsions, and coma. Type 1 addiction usually starts after age 25, and it's often associated with life stress or peer pressure. And type 2 addiction is usually apparent before age 25 and is thought to be hereditary. There's a strong genetic component. Okay, how do we treat this? Well, there's behavioral modification. Examples would be abstinence, peer support, avoidance, or correction of stress, which can encourage drinking, as well as psychotherapy. And there's also compounds that can suppress the desire to take in alcohol. One example would be antabuse, which results in severe nausea if the individual takes in alcohol while on this particular drug. However, in order for it to be effective, you have to have patient compliance, which is sometimes a challenge. So this is not a cure-all by any means, but it is one approach to treating addiction. So with that in mind, we've reached the end of this particular presentation, and I hope this finds you well. Thank you very much.